Afternoon to everybody present here. We are here to participate in a webinar series launched by the Association
Hello.
of Microbiologists of India. Am I audible? Microbiologists of India. This
Yes,
lecture
ma'am.
series is an effort to Association of Microbiologists in India to acknowledge and appreciate the tremendous
Okay, so I have
impact
now
and
another problem.
achievement in the field of microbiology and having our could have and various aspects of our lives. The use of microbiology is believed to have a potential to revolutionize a wide range of scientific and biological fields. This series of lectures will make close association and the scientific connection with the scientists, educationists, professionals, technocrats, and the students during this lockdown period or pandemic situation. This webinar series is great effort
I
of
cannot hear
Association of
anything.
Microbiologists of India to keep the knowledge update and utilize time best possible way in the current scenario in this pandemic situation. This webinar has received an overwhelming response from the scientific community. In fact, we have 380 registered participants. Other than this, we will be also broadcasting online session on AMI Facebook and AMI YouTube channel. Today, we have with us Professor
Hello?
Neelam Sangwan. Yes, hello. Am I audible or not? Hello.
Audible, ma'am.
Okay, but somebody is complaining that I'm not audible. That's why.
And the speaker, sir, is asking because he could not hear your sound.
Yes.
Okay. Professor Neelam Sangwan. She is the head of the department, Biochemistry Division, Central University of Mahendragarh or Haryana. She is a well-known molecular biologist and recipient of many national and international awards. Likewise, K.S. Krishnan Fellowship, Department of Atomic Energy in 1988, INSA Young Scientist Medal in 1994, DST Boykoss Fellowship, ISKA Professor Hiralal Chakravarti Award in the 2007, CSIR Technology Award in Life Sciences 2015. Women leader in crop science and agriculture, member delegates under Newton Bhava program, Cambridge University, Cambridge, UK, in 2016. I welcome you, Madam. On behalf of the Association of Microbiologists of India,
Okay.
as General Secretary, I take this opportunity to accord a very warm welcome to our today's speaker, Dr. Namit Ranjan, Research Scientist, Department
So
of Physical
my computer
Biochemistry,
is hanged or what?
Max Planck Institute for Biophysical Chemistry, Gottingen, Germany. On this technical session of Dr. Namit did his PhD from Zurich, Germany in the field of molecular biology and biophysics in 2010 and master from the Madurai Kamraj University in 2006. Dr. Namit secured All India second rank in MSc entrance examination for getting admission in the biotechnology program. He started his postdoc fellowship in the Max Planck Fellowship, Molecular Biology Division, and topic of his today's lecture on translational regulation in the eukaryotes, the role of regulatory network and RNA API transcriptome. Very relevant to today's scientific development in the field of molecular biology. I have a great pleasure to welcome you, sir, in this wonderful virtual gathering. Now, I request Dr. Namit, please share your screen. Thank you very much.
Okay.
My request to everybody to please don't. Now, over to Dr. Namit Ranjan, please.
Okay. So Thank you very much for this kind invitation and introduction, and I'm very sorry for the problem that has been created now. So I hope everyone can see my first slide, and then I can get going. Is that right, Professor Singh?
Yeah.
Can you see my slide?
Yeah,
Okay,
yeah,
good.
yeah, that's perfect.
Okay, good, perfect. Thank you. Okay, so today I'm going to talk about how translation, the basic cellular fundamental process, is regulated not only in eukaryotes but in all domains of life. And specifically, I'm going to talk about the role of RNA epitranscriptome, that is the tRNA modification, and the role of regulatory network. So as we all know, translation is a very basic process, which is basically involved in 4 major steps called as initiation, elongation, termination, and recycling. And during initiation, the programmed ribosome is ready for elongation to happen, where the elongation is kind of subdivided into 2 steps. One is called as decoding, where amino acid tRNA comes and goes inside the ribosome, and a peptide bond is formed. And once a peptide bond is formed, the ribosome has to move along the mRNA, making another codon to come inside the ribosome. And this process is called as translocation, the movement of ribosome and the tRNA along the mRNA. And this is facilitated by a special elongation factor, EFG, in bacteria. And there are 2 elongation factors, EF2 and EF3 in yeast, which is required for this translocation to happen. And this process of decoding and translocation continues until the ribosome encounters the stop codon at the A site of the ribosome, and where the termination happens in presence of release factor. And after the peptide bond is released from the ribosome, the ribosomal subunits, the mRNA and the tRNA are kind of recycled through ribosome recycling factor. And all these ribosomal subunits can re-enter the process of translation through initiation. So of course, this process is very well conserved throughout all domains of life. However, we do have significant difference in the number of translation factors that is required. As you can see, in bacteria, we have only 3 initiation factors, whereas in eukaryotes, we have more than 5 initiation factors. And that's also true for some of the elongation and release factors. So what I want you to focus today is that there are 2 important factors which I'm going to talk today about. One is the role of tRNA. So far, people always thought that tRNA is a passive molecule whose business is to bring the amino acid tRNA inside the ribosome, and the ribosome takes over the process. So today I'm going to show data and convince you that tRNA is just not a passive molecule, but it's a very active molecule, and it does regulate translation. Okay. And the second talk that I'm going to tell you, a story that I'm going to tell you today to you all about, is a yeast-specific elongation factor called EEF3. It's an essential elongation factor in yeast. And today I will tell you what does it actually do inside in translation and how it regulates translation. So let me start with the first part, which is RNA epitranscriptome. So this terminology, epitranscriptome, is defined as a post-transcriptional alteration that do not change the RNA sequence per se, but they are functionally relevant for gene expression. So one can imagine these kind of RNAs would be then tRNA, the mRNA, and the all non-coding RNAs that are present in the cell. So by now we know that there are lots and lots of modification that has been detected in RNA, especially tRNA, rRNA, and very recently in the mRNA. So I will tell you about the tRNA, which is a very highly modified species. As you can see here, even this figure is not up to date. It's actually 10 years old figure, and now we have more than 150 tRNA modifications present in all across all the tRNA species. And one of the hotspots that is very obvious to see is position number 34, which is the first anticodon position on the tRNA. This is highly modified in all, basically essentially all tRNAs. Okay. And today I will focus more on this position 34 and how it can regulate translation. And the modification that I'm gonna talk to you all about is Mcm5s2u modification, and I will tell you what this short form means and what it does. But before I go into that, the question is why to study tRNA? So we all know, okay, tRNA brings the amino acid inside the ribosome based on what is the codon at the A site, and that's it. But over a number of years, by now we know that there are numerous human diseases associated with tRNA modification, and some of the diseases are listed over here. It spans through neurological disorders to cardiac disorders to respiratory disorders to cancer to metabolic disorders like diabetes, and also mitochondrial linked diseases. So it kinds of spans all kinds of major diseases that anyone can have in their lifespan. Therefore, it becomes very, very essential in addition to all other factors and the processes that we are studying to understand the process of disease, that how tRNA, which is a very small molecule, can in principle regulate translation. And if that goes wrong, then how this can lead to disease. Okay. So this was all the motivation by which we started to study the tRNA modification. So as I mentioned before, that I will talk to you all about Mcm5s2u modification. So Mcm5 stands for methoxyl carbonyl methyl, and S is thio modification. So this is present at the 34th position of 3 tRNA, which is lysine, glutamine, and glutamate. And the methyl modification is actually done by, in yeast, is done by ELK pathway, which is composed of more than 6 various proteins, and they all come together to do the methyl modification at 34th position, at the 5th position of actually uridine in these 3 tRNAs. On top of that, position number 2 is actually modified by a thiosulfur group. And by now we know that this is done by a URM1 pathway, which is composed of at least 5 various proteins. And the pathway is kind of very complex, how this thio is placed on the tRNA. So I will tell you today if there is thio present on the tRNA, or if sulfur is missing on the tRNA, how it can regulate translation. But before that, in the literature, people always kind of predicted that it can restrict and facilitate Wobble-based pairing, just because position 34 interacts with the 3rd position of the mRNA, which is the Wobble position. So this was a kind of a very obvious hypothesis to say that it can restrict or facilitate Wobble-based pairing, and therefore this can fine-tune decoding at the ribosome. But there was no concrete evidence to show that this is actually really happening. So we started to understand how is translation affected. This was done a couple of years back, and we started to understand how if you delete a gene which is responsible for thiolating the tRNA, in this particular case I'm talking about URM1, if you delete URM1 in the yeast cell, which is a non-essential gene, you end up with a non-thiolated tRNA. Okay. So we compared the polysome analysis of fully wild type cell, where we will have all the modified tRNAs, compared to URM1 deleted cells where we don't have thiolated tRNAs. So we could not see much difference between the polysome analysis, and therefore we decided to do a proteomics analysis, because this would give an indication that if there is no difference in polysome analysis, that means most of the proteins are kind of unaffected, and there is only handful of substrates or proteins that is required for the translation of those proteins. The fully thiolated tRNAs are required. So when we performed the proteomics analysis, we could see that there are a bunch of proteins which are down-regulated if you delete the URM1, or in presence of non-thiolated tRNAs. And of course, there are a bunch of proteins which are upregulated. Since we were interested to understand how protein translation can be down-regulated by the absence of sulfur group on the tRNA, so we mostly focused on down-regulated proteins over here. And as I've shown you, that there are 3 codons which require thiolated tRNA for lysine, glutamine, and glutamic acid. So we asked, out of these proteins and basically genes, how many of them are enriched for these 3 codons which would require a fully modified thiolated tRNA. And indeed, we could only find a few of them. This kind of proof that the polysome analysis is not affected, because we only have a handful of genes that requires a fully modified tRNA for its proper translation. So in addition to that, we have done number of control experiments to make sure that the mRNA level of these proteins or genes, what is shown over here as black dots, remains unaffected in wild type cells or all in the mutant cells where we expect no thiolation of tRNA to happen. Then the next question we asked was, okay, if the mRNA levels are same, but the protein levels go down, does it because of the protein stability? So it could still happen that the unmodified tRNA results in a translation and the product is kind of unstable and would be degraded very fast. So what you see here is only 2 examples out of all these. However, we have tested all the black dots, or basically all genes and protein products shown here, and we could not see any difference in the half-life of the protein, whether it's from the wild type or from the unmodified tRNA background cells, okay, suggesting that the effect of downregulation that we see is actually happening on the level of translation or inside the ribosome, okay. So from here we concluded that general translation is not affected, and there are only a handful of genes which are enriched for these 3 codons. They would require a fully modified tRNA for proper translation to happen. So when we published this result, so essentially one result in the same year and one result from the lab of Sebastian Leidel in 2 years later came to the same conclusion, where what they have done is they have performed a ribosome profiling assays or experiments where they could show that if you have a fully modified tRNA or fully thiolated tRNA, you have an optimal codon translation rate, and therefore you will have a proper folded proteome. However, when you have an unmodified tRNA or the thiolation or sulfur group is absent on the tRNA at 30-fold position, you have a codon-specific ribosome pausing, and these codons are the codons which would require fully modified tRNA. And at that point, they call it as ribosome profiling, and I would show that pausing, and I would show that the ribosome actually does not pause, rather it moves very, very slowly on these codons, okay? And if you have a ribosome slowdown, this could result in protein misfolding and aggregation. And from their ribosome profiling experiments, what they could observe is that when they do the profiling experiments from a unmodified cell background, they could find about 30 to 40% more ribosomes on these codons compared to wild type. So meaning they have 30% ribosomes more they have captured, okay? So this information we should keep in our mind, because I will convince you and show you that how this 30% to 40% of extra ribosomes on these codons can happen, okay? So after that, I came to the lab of Marina Rodnina here at the Max Planck Institute, and we started to understand how is translation regulated. So Rodnina lab is a very well-known lab to understand the regulation of bacterial translation or prokaryotic translation, and I joined the team to understand how the eukaryotic translation is regulated, and starting with the focus on tRNA modification. So before I go into details, I have to first walk you all through the A site binding scheme, or basically the process, how the decoding happens on the ribosome. So after the initiation complex is formed, meaning initiated tRNA comes at the P site where AUG codon is there, and then the A site, in this particular case, in our experiment, we had lysine codon. That's why you see AAA written over here. This A site is free, and this programmed ribosome is ready for decoding to happen. And for this process, amino acyl tRNA, which is shown in blue here, in complex with an elongation factor in its GTP bound form, come and initially binds to the ribosome with some on rate which we call as K1 and an off rate which we call as K-1. And after initial binding, there is a step called codon reading and codon recognition, by which the ribosome recognizes, based on the codon that is present at the A site, which of the ternary complex is suitable over here, and then the proper tRNA goes inside the ribosome. That means the codon is recognized, and this step is called the rate on rate, we call it as K2, and the off rate is called as K-2 here. Once there is a proper codon recognition, then the GTP of elongation factor is activated and hydrolyzed. And with the hydrolysis of GTP, the inorganic pyrophosphate has to leave the elongation factor, and till this step is called as initial selection, because after GTP is hydrolyzed, there is no way the tRNA will be dropped off based on codon recognition and initial binding. So once the PI is released, EFTU undergoes a rearrangement, or the elongation factor undergoes a rearrangement, and still, if ribosome believes or senses that this tRNA is not proper, if there is wrong tRNA that has come inside the ribosome by chance, or if there is no proper codon-anticodon interaction of the tRNA with an mRNA, then the amino acid tRNA will be rejected from the ribosome without forming a peptide bond formation. Okay. Now, if everything is fine, if the tRNA is proper and ribosome senses that this tRNA is fine, then the peptide bond is formed. And before we could publish our story, there were numerous X-ray crystallographic structure or cryo-EM structure suggesting that the peptide bond formation can happen in presence or absence of elongation factor. That means there's two ways by which a peptide bond is formed. One way is which is assisted by elongation factor in its GDP bound form for a peptide bond to happen before it can leave the ribosome. Okay. And the second possibility is that EF2 will release, the elongation factor is released, and the peptide bond is formed on its own, resulting in a dipeptide bond formation. However, we still don't know which pathway is preferred over the other. However, our kinetic experiment and the structural experiment concretely show that these two pathways are essentially possible. Okay. So now, my aim was to understand how each and every step, what I have shown here, is regulated by unmodified and fully modified tRNA over here. That means I have to design and experiments to monitor each and every step what is written over here. So now I will walk you through how I did that. So to understand the initial binding step, what we have done is we have labeled, fluorescently labeled our body of the tRNA with proflavin. And once the proflavinated amino acid tRNA interacts with the ribosome, we would expect an increase in the signal because it gets quenched and there is an increase in the signal. And this would suggest that the amino acid tRNA or the ternary complex interacts with the ribosome. Okay. Now, for codon recognition and also to get some information about peptide bond formation and amino acid tRNA rejection, we use the same proflavinated tRNA. However, to perform codon, these kind of experiments, we have used programmed ribosome. And for initial binding experiments, we have used empty ATS ribosome. Otherwise, we would not differentiate between initial binding and codon recognition experiments. Okay? So the difference between initial binding and codon recognition is empty ATS ribosome versus fully programmed ribosome with an initiated tRNA at the P site of the ribosome. To follow and monitor GTPase activation, we have used the fluorescently labeled GTP, marked GTP. And once the GTP is activated, you see an increase in the signal. And then once it's hydrolyzed and it's released from the ribosome, you see a decrease in the signal because this would mean that it is leaving the ribosome after the job is done. Now, to follow the GTP hydrolysis, we have used basically took advantage of radioactivity, where we have gamma phosphate labeled with 32P of GTP. And once this is hydrolyzed, we would see an increase in the release of PI and GDP formation. So this increase in this radioactivity would suggest that the GTP is hydrolyzed over time once the ternary complex goes inside the ribosome. And for peptide bond formation, again, we used radioactivity where our amino acid was labeled with an 14C amino acid, radio labeled amino acid. And then once a peptide bond is formed, we would expect to see an increase in the dipeptide fractions or tripeptide fractions, whatever we are interested to study over time. And so once I have established all these ways to monitor each and every step, so then I collected time-dependent traces for all the steps, basically initial binding, codon recognition, GTP activation, GTP hydrolysis, and peptide bond formation using fully modified tRNA from wild type, or for using non-thiolated tRNA from the mutant cells. Okay, now here it's very difficult to understand what is happening, except we see that there are lots of traces, which essentially does not give any information about the rates of initial binding and everything. So in order to deduce the elemental rates of each and every step, we have used Kintek Global Kinetic Explorer. It's commercially available, and it's one of the let's say latest software that we use to understand our elemental rates. And to cut down the whole story short, I'm going to show you what are the essential steps that are regulated by the presence or absence of sulfur on a tRNA. Now, sulfur is a very small molecule. However, it can regulate translation on a very global level. So initial binding, and what you would see here is the black trace is always from, or the black bars are always from the wild type fully modified tRNA, and the blue bars are always from the unmodified tRNA. Now, for initial binding, the rate of initial binding or the rejection from the ribosome has not affected in presence or absence of sulfur group on the tRNA. And this was kind of expected, because if there is anything has to happen, it can only happen once the tRNA goes inside the ribosome. Therefore, we were expecting to see some difference in the codon recognition step. So the way ribosome senses the tRNA is similar. So it does not discriminate whether the tRNA is modified or not modified while the tRNA is going inside the ribosome. Once the tRNA is inside the ribosome, then the ribosome senses that, oh, this tRNA is not fully modified, therefore it should go outside the ribosome. And that is the rate of rejection from the ribosome, the codon rejection. And this is 6 times higher or faster than the fully modified tRNA. So that's the first step, where ribosome tells the tRNA that you're not fully modified, therefore you should not come inside, because we will have problems in making peptide bond formation. But of course, there are a certain amount of tRNAs, because in cells, everything is so crowded that the ribosome does not have enough time to tell all the tRNA to be rejected and to halt or pause the translation. So there's always a chance that some tRNAs, unmodified tRNAs, still can go inside the ribosome and a peptide bond is formed. And GTP activation and hydrolysis, the rates are so fast that we could not see any significant difference in the by the presence or absence of unmodified and unmodified tRNA. The surprising thing came to us when we realized that the PI release, or monitored the PI release and the EFTU rearrangement, that from the unmodified tRNA, for some unknown mechanism that we still don't understand, the release of inorganic phosphate is very, very slow, about 7 times slower than the fully modified tRNA. And so this is the second step, where the ribosome kind of senses that there is a problem of tRNA not being fully modified, and whatever process happens afterwards is very, very slow. And we could not see any significant difference between the peptide bond formation, whether it is EFT elongation factor dependent process or an independent process of elongation factor. Then the another important step that we could see there is a problem if you don't have fully modified tRNA is the tRNA rejection step. So of whatever tRNA passes the codon recognition step, passes the PI release and elongation factor rearrangement step, can still be rejected from the ribosome without making a peptide bond formation. And this was 3 times faster than fully modified tRNA. So at the end, we could figure out that in the whole process of decoding, which is a multi-step process, there are 3 intermediate steps, which is kind of regulated by the absence of thiolation on the tRNA. So well and good, whatever tRNA manages to make a peptide bond formation, then after decoding has happened, then the ribosome has to undergo a process called as translocation, where ribosome and the tRNA moves along the mRNA to the next codon. So this process is facilitated in bacteria by EFG in its GTP bound form. It's called as a translocation factor. And once the translocation factor binds to the ribosome, then the ribosome undergoes a counterclockwise rotation, which allows tRNA to move to the next codon. And once this step is done, then ribosome goes back to its original position called as clockwise rotation. Okay, and by this, the ribosome and the tRNA has moved one codon to the right towards the next codon on the mRNA. And in this way, the E site tRNA is released, and then the tRNA, which was at the A site, comes to the P site, and therefore making A site free for the next round of decoding to happen. So we were interested to understand if the process of decoding is slow, does it also affect the process of translocation? So whether the modified tRNA makes the translocation process faster than an unmodified tRNA. So we monitored two particular steps here. We have labeled our ribosomal subunit with a fluorescent tag to monitor the FRET observation of the counterclockwise rotation of the ribosomal subunits and the clockwise rotation. And then we performed another assay, which is called as Pyromycin assay. So Pyromycin is a very small chemical molecule which can bind to the ribosome as soon as there is a free A site in absence of any ternary complex. So if the translocation is fast, then you have faster exposure to the A site, and therefore pyromycin reaction will be faster. So that was the idea. So when we monitored the FRET observation of, or the relative fluorescence of the ribosomal subunit movement, we could see that for fully modified tRNA, the counterclockwise rotation or the clockwise rotation of the ribosomal subunit was not affected. Okay, and for the unmodified tRNA, the counterclockwise rotation of the ribosome was not affected, but the movement of the tRNA from A to P site and P to A site was slow, and therefore we could observe a slow clockwise movement of the ribosome, resulting in a native formation of the conformation of the ribosome, which was about 2.5 times slower than fully modified tRNA. Now, if the rate of the movement of tRNA and ribosome is 2.5 times slower, meaning this would make an A site free 2.5 times slower. Therefore, we expect a 2.5 times slower pyromycin reaction, and this is precisely what we have seen. So it's very clear from here that the decoding is slow. Not only decoding, but the translocation of the ribosomal translation is actually also affected by the presence or absence of tRNA modification. Now we have all the elemental rates. So we asked our Kintech Explorer, can you please calculate for us the decoding and the ribosome residence time, basically translocation, from the elemental rate, and tell us how slow the decoding would be from a fully modified tRNA to an unmodified tRNA. And we got a number that for fully modified tRNA, the decoding is 1 second basically, and it moves from 1 second to 3 seconds when you have an unmodified tRNA present at the A site of the ribosome. And for translocation, we have calculated 2.5 seconds for ribosomes to sit there for fully modified tRNA, and this has moved to 3.5 seconds for the unmodified tRNA. And from there, we can expect an increase of the ribosome residence time on that particular codon for about 40%. Now, remember from the ribosome profiling experiments, they have seen about 30 to 40% more ribosomes captured on the codons that would require fully modified tRNA, and this, our in vitro kinetic experiment, also came to the same number. So meaning and suggesting that this is one of the let's say very few chances where both in vivo and in vitro experiments come together and give us the same conclusion. So from there, we have concluded that actually ribosomes are not paused, rather they are moving very slowly, because the decoding is slow, the translocation is slow. Therefore, whenever you capture the ribosome, you have 40% more chance to capture ribosomes on these codons if you are capturing the ribosomes from the mutant cells, which does not have fully modified tRNA. So So in summary, what we have shown is that if you have fully modified tRNA, you have very fast decoding, you have very fast translocation, and therefore you have an efficient translation. The moment you lose thio group on the tRNA, you have a very slow decoding, you have problems in translocation, so you have very slow translocation, and thus resulting in slow translation. And this is what results in less protein amount that we have observed in our proteomics data. Okay, so from here we can conclude that tRNAs are actually not a passive molecule. It's really a very active molecule which very actively regulates translation. Okay. Now, of course, this is the story for the tRNA modification. We are also studying the modification on internal mRNA, so basically in the coding region of the mRNA, which was identified like 6 years back now. Of course, we know M7G cap, which is at the 5' end of the mRNA, but recently people have identified the modifications within the coding region of the mRNA, and we don't understand why this could be. So we are, of course, studying all those things, but and this is also part of RNA epitranscriptome, but I will not have time today to talk to more about that. So from here on, I'm gonna move to the other part, which is the regulatory partners of ribosome. So as I mentioned that in yeast, we have a eukaryotic elongation factor 3 or EF3, and this is an essential gene in yeast. So the moment you delete the gene, the cells are dead. So far, it's very fungal specific. There's no known homologs found in any other organisms. And it contains 2 ATP binding sites called as ABC1 and ABC2. Both are shown to bind and hydrolyze ATP. And of course, since it's an elongation factor, it is known to be involved in translation, elongation, and termination. However, there is no precise role known in exactly what step it is involved and how it can be mechanistically regulate translation. Now, if we look at our yeast genome database, we could also see that elongation factor 3 controls over the rate of translation. Its expression is changed upon starvation and morphogenesis, and interaction with the ribosome is attenuated by another stress-related factor called as STM1. And it interacts with various translation factor and other factors, and the role of these interactions are still very much unclear. And therefore, the precise role of EF3 remains elusive. I should stress here that there's one crystal structure available from 2006, which could show that EF3 kinds of binds to the both subunit of the ribosome, both the 40th subunit and 60th subunit of yeast, and it binds towards the E side, and therefore people thought that it must be involved in termination as well. Okay, now in order to understand the real mechanism of this function of EF3, we cannot do anything in vivo in detail just because it's an essential gene. So in order to study the mechanism of this one would really require to have an in vitro translation system, and this is what we have done. So we have fully reconstituted in vitro yeast translation system where we have purified all, not all, however, the minimum initiation factors, elongation factors, the ribosomal subunit that is required to reconstitute our ATS initiation complex. And this system is very powerful because you have a possibility to add and delete whatever factors you are interested to study to reconstitute your ATS initiation complex, and therefore monitor how initiation is regulated, and also to monitor each and every step of elongation, as I have shown before using bacterial ribosomes here. Okay, now this is of course a very powerful system. So what I'm going to show you here is our purification gels of all initiation factors, all elongation factors, the ribosomal subunits that we have done. Some of the initiation factors are purified from native source, directly from yeast. They are denoted by native names over here. All elongation factors are purified from yeast cells. Of course, the ribosomal subunits are also purified from yeast cells. Now, well and good that we have all the factors purified and present in our hand. The next question was, whether we could reconstitute the yeast translation system. So what we have done is we have prepared our ternary complex, which is a complex of EIF2 in its GTP-bound form, and an initiated tRNA, which is amino-acylated with a radio-labeled methionine here. And then we add all the rest initiation factors, the ribosomal subunit, the mRNA, the 60S subunit, and monitor the formation of ATS initiation complex on a size exclusion chromatography. Now, if this ATS initiation complex is formed, then we would expect the A260 for ribosome and radioactivity coinciding within the same peak. And this is precisely what you see here. The black trace is the absorbance of A260 for the ribosome, basically the RNA from the ribosome. And the blue bars that you see here is the radioactivity count. So the first peak is where ATS initiation complex elutes. Now, we have used excess of initiation factors that comes out of the, that does not remains with the ATS initiation complex. And then this is the excess radioactivity of initiated tRNA that we have used to reconstitute the system. So very good, bingo, we have our ATS initiation complex formed. Then the question was whether they are active. Can it make decoding? Can it do the job it is supposed to do? And indeed it could do. So what we have done is we have taken our ATS initiation complex, we added the elongation factor associated with its amino acid tRNA corresponding to the codon that was present at the A site here. And in this particular case, what you see is the formation of MET-Val, the dipeptide. And as you can nicely appreciate that with time, there is a formation of product suggesting that our ATS initiation complex is active. Now, this thing we have assembled and put together into a book chapter, which is supposed, so by next month, it should be available for everyone to read and reconstitute if they want. So now, well and good, we have all system ready. We have our initiation, ATS initiation complex ready. We have all the elongation factors ready. So now is the time to really see what is the role of EF3 in translation and how it can regulate translation. So the first set of experiments we did was we monitored the formation of dipeptide, Met-Phi in this particular case. And from now on, the Met-Phi or the dipeptide complex, I would call it as ATS2C. So in order to monitor formation of ATS2C, we have used our ATS initiation complex. We mixed it with our ternary complex, which is elongation factor in its GTP-bound form, and the amino acid tRNA corresponding to Phi codon at the A site, and monitored the formation of dipeptide over time. And just for your information, we are using this mRNA, which don't have a stop codon. Therefore, we have possibility to, in fact, go further in our translation system. So we have performed the formation of dipeptide in presence of EF3 or in absence of EF3. And as you can see that the presence or absence of EF3 essentially does not affect the formation of dipeptide. And therefore, from here, we have concluded that the dipeptide formation is independent of elongation factor 3. And this would kind of make sense because the name itself suggests that it's an elongation factor and it's not a decoding factor. Therefore, this kind of result is very promising and very much expected. So then the next thing was to understand how EF3 is playing a role in tripeptide formation. So as I showed you that the first peptide bond formation or dipeptide formation is independent of EF3, and after a peptide bond is formed, as we know that a process of translocation has to happen to make A site free, and then another ternary complex corresponding to the codon at the A site would come and a tripeptide is formed. So we monitored how the tripeptide formation is kind of regulated in presence or absence of elongation factor 3. So as you see here that we have mixed our dipeptide complex with ternary complex. In this particular case, the ternary complex was with valine tRNA, and we have mixed our ternary complex with both factors EF2 and EF3, because EF2 is known translocation factor in eukaryotes and EF3 is an essential specific yeast elongation factor. So we have mixed either with both elongation factors or with EF2 alone or with elongation factor 3 alone. Now, when you add or monitor the tripeptide formation in absence of one or the other elongation factor, you don't see any formation of tripeptide. But the tripeptide is only formed when you have both elongation factors present, both EF2 and EF3. So this strongly suggests that EF3 is really, really required for tripeptide bond formation. Now, the question remained whether this is happening at the level of translocation or this is happening after translocation. And I will going to talk to you about that as well. Now, if EF3 is really required for tripeptide formation, then what we have done is, as a control experiments, we have performed our ATS-IC. We have mixed with the elongation factors, EF2 and EF3, or with EF2 or with EF3 for about 15 minutes before mixing the ternary complex Val. Okay. So now, and here what we have see that the EF2 alone, the red triangles, so in 15 minutes, EF2 can alone allow translocation to happen so that A site is free and a tripeptide bond formation can happen, similar to the presence of EF2 and EF3. And EF3 alone can also tries to help ribosome to translocate. However, it does not do to the same extent as EF2 and EF3 can do. So now it's very clear that in absence of EF3, EF2 alone can do, but it can do the business or the translocation very, very slow compared to the presence of EF3. So meaning EF3 kind of facilitates the formation of tripeptide or enhances the tripeptide formation. So then we moved one step back to understand what is the role of EF3 on translocation. And as I've shown before, that in the process of translocation is subdivided into two steps. One is the counterclockwise rotation, which allows the tRNA to move from P to E and A to P side, and moving back to the original form of ribosome, where A site is now free for next round of decoding to happen. So we monitored the Pyromycin assay, which would allow and tell us how fast this A site is free so that the pyromycin can react. So again, here we have mixed our ATS dipeptide complex with EF2, EF3, and pyromycin, or with EF3 alone or with EF2 alone. Now you can see that compared to presence of both factors, the presence of elongation factor 2 alone is sufficient for a pyromycin to react. So now this means that EF2, this can only happen if you have a counterclockwise rotation of the ribosome happen. So as soon as the counterclockwise rotation of ribosome happens, the pyromycin can go and interact with the peptidyl tRNA and make a MET-Phe pyromycin. So this suggests that EF2 is the first factor which is involved in counterclockwise rotation of the ribosome. And this is not done by EF3 because EF3 cannot reach to the same level as the presence of EF2 alone here. So we concluded that EF2 kind of do the counterclockwise rotation of the ribosome, allowing the tRNA to move, and the closure of the domain is kind of involved or responsible factor to do that is EF3. And I will show you that this is the case indeed. So as a control experiments, we again performed where we mixed our dipeptide complexes with EF2, EF3, and the pyromycin, or with EF2 alone or with EF3 alone. Okay. Now you see that indeed the presence of EF2 alone is sufficient, whether it is in pre-translocation complex or in post-translocation complex, very strongly suggesting that EF2 is the first translocation factor which kind of makes the counterclockwise movement of the ribosome, and EF3 plays a role at the later stage of translocation. Because here, if EF2 is not present, the pyromycin cannot react with the dipeptide complex. So very strongly suggesting that EF2 is the first translocation factor and EF3 is the second translocation factor. So now, after there, we asked whether EF3 has any role in E-site clearance, because the crystal structure and the cryo-EM structure that we have shown in our manuscript now also shows that it binds towards the E-site. So in order to understand the role of EF3 in E-site clearance, what we have done is we have followed the dissociation of tRNA, which is fluorescently labeled at the body of the tRNA from our dipeptide complex. So the blue tRNA here would mean the fluorescein tRNA. So the label is on the blue tRNA here, and then we monitor the release of the tRNA, because in presence of EF3 or in absence of EF3. So what we see is that when we mix our dipeptide complex with just single initial elongation factor, whether it's EF3 in blue or EF2 in red, the release of or dissociation of methionine tRNA from E site is very, very slow compared to when you add both factors, EF2 and EF3. So here, in presence of EF2 and EF3 would mean one round of translocation. That means P site tRNA goes to E site and A site tRNA comes to P site. So the presence of EF2 and EF3 releases the E site tRNA in first round of translocation in absence of ternary complex. And when you allow tripeptide formation to happen, that means it can undergo 2 rounds of translocation, because in first round some of the tRNAs are released, in second round tripeptide is formed, and then this red tRNA comes to the E site and the blue tRNA moves from A site to P site. This also happens because you have EF2 and EF3 present in your solution. So what we have observed is that for complete clearance of E site tRNA, you need 2 rounds of translocation and a tripeptide bond formation to happen. Okay. Now, what we have observed is that when we pre-incubate our ATS dipeptide complex with just EF2, that means only the counterclockwise rotation has happened. The tRNA does not, the E site tRNA does not leave the ribosome. Okay, so meaning the E site tRNA remains inside. So all 3 tRNAs are there on the ribosome, and in order for E site tRNA to release the ribosome, EF3 is really, really required. Okay, so we conclude that EF3 promotes the dissociation of deacylated tRNA from the E site of the ribosome. So what I'm not going to show here is that we have confirmed using ribosome profiling with our collaborator and cryo-EM structure again from our collaborator, which shows very precisely that the E site tRNA remains inside the ribosome in absence of EF3. So EF3 is really required for complete translocation to happen so that the E site tRNA can be released from the ribosome. Okay. So let me now summarize what we have shown and studied so far. So we have shown that EF3 does not affect the first peptide bond formation. Rather, EF3 facilitates the tri and polypeptide bond formation, and EF3 facilitates the late stages of translocation, because in absence of EF3, there is no tripeptide bond formation, there is no E site tRNA release from the ribosome. And EF3 promotes the dissociation of deacyl tRNA from the E site. Okay. So with this, I'm gonna now conclude my talk, and the take-home message is that actually EF3 is not an elongation factor, it's rather a proper and a very precise translocation factor, which is responsible for completion of translocation to happen. And if translocation is not complete, the ribosome cannot do translation, and therefore this is very, very important and essential factor in yeast cells. And of course, we and others are also trying to figure out and hunt if there is an homologue of EF3 present in other higher eukaryotes, because it makes kind of very, very dicey situation that EF3 is only present in yeast so far and not in any other organisms. So with this, I would like to acknowledge a couple of people. So the first is Matthias Peter at ETH Zurich, where the whole tRNA modification story started. And special thanks to Patrick Pedrioli, who did the proteomics analysis for us. And then very, very special thanks to my current advisor, Marina, for giving me a very fantastic opportunity not to only continue the RNA epitranscriptome journey, but also to give me freedom to establish the whole translation system of East, and to do this in a fantastic journey with support from a postdoc in the group Sandra Blanchet. Sakshi Jain is a graduate student who is working on mRNA modification, and Tessa Hübner is a very talented technician in the group, in our East group, who is handling and taking care single-handedly to maintain each and every stock of all the initiation factors, elongation factors, and everything that is required for our East translation system. Very special thanks to the DFG, or the German Science Foundation Priority Program 1784, which gave my first independent funding to study mRNA modification, the Max Planck Society for their generous support, and also our institute, Max Planck Institute for Biophysical Chemistry, for their constant support. So with this, I'm going to thank you all, and I will be very happy to take questions from you all. Thank you.
Wonderful talk, sir.
Thank you very much.
I'm thankful to you. Dr. Namita has left somehow, and she will be soon joining.
No problem.
Dr. Anil Panghal, if you are there, kindly ask the queries, if any. Dr. Anil Panghal.
Thank you, Doctor. Thank you, Dr. Nilesh.
Thank you very much, sir.
Thanks, sir, for the nice presentation and very wonderful presentation you have given.
Thank you.
Some questions are there, sir. If you allow me to take those questions, I can ask those questions based on the behalf of all participants.
Yes, please. Is some disruption there? I cannot hear.
Actually, sir, I'm searching for those questions.
Okay, sorry.
Dr. Namita Singh had to ask these questions as she had left due to some technical problem.
No problem.
So I had to ask those questions.
Sure.
So one question is from Mrs. Prerna, Ms. Prerna Jalkote, sorry. How did you do or perform the fluorescence labeling of subunits of RNA?
Okay.
Which fluorescence molecules actually used?
Yeah. So this method of labeling ribosome subunits was actually established in a group in US, and we have used Cy3 and Cy5 to label the one of the ribosomal proteins. So we purified that protein individually, labeled them individually, and reconstituted with the rest of the ribosome, which was purified from the cells lacking that gene for that particular protein that we have labeled. So we have, let's say, U11 proteins, the large subunit protein in number 11. So we have deleted that gene from bacteria, and purified the ribosome lacking large protein 11, and then individually we have purified L11 protein and labeled it and reconstituted to make the large subunit of ribosome. And the same thing was done for the small subunit, and that we have used. So it's a very, I would say, fantastic way to label the ribosomes. And we have also confirmed that the activity of ribosomes are not deteriorated or has not gone down by making sure that they are active to make the peptide bond formation. Okay. And if you are really interested to understand and know the protocol, it's of course mentioned in our manuscript, and I will be very happy to share that as well with you all. So we have purified individual protein, labeled it individually, and for the rest of the ribosomes, we have purified from the mutant cells and reconstituted together.
Thank you, sir. Another question is from Megharaj Kaviraj. Megha Kaviraj, sorry.
Yeah.
And please explain the leucine codons used at about the same frequencies in different species. For example, in humans and E. coli cells. Is there any bias in codon uses related to the AT by GC content? of the genomes of different species?
So yes, of course, there are certain biases towards certain codons. For example, in bacteria, there are certain codons that are fastly translated in the initial part of the mRNA, and then it kind of changes with eukaryotes and also to higher eukaryotes. So so far we don't understand why this would be the case. And one possible reason is the regulatory network of ribosome is so tight. So I only talked about or showed you on the known translation factors that are involved to interact with ribosome and how it can regulate it. But we have numerous other translation ribosome interacting partners which only interacts for certain parts on the mRNA and then goes off the ribosome. So there are certain biasness towards that one. And in terms of RNA modification, so the tRNA modification is kind of very independent. For mRNA modification, the mRNA can be modified at anywhere, essentially anywhere, because so far we don't know if there is a signature sequence. If that is present, then only a certain modification will happen, or under stress, if there is a different signature motif. This we don't know. So essentially, for now, any mRNA base can be modified. And the well-known study is M6A. So the adenosine at the 6th position, the methyl modification can happen essentially in any A's present on the mRNA randomly. Okay. But it's very unlikely that this is going to be the case. But we have to just wait for people to come up with certain explanations. So we are also running behind that to understand and basically address the codon biasness towards that. Yeah.
Thank you, Dr. Namit. Now I request Professor Neelam Sangwan ma'am for the concluding remark of today's lecture.
Thank you very much. First of all, I thank AMI, I thank Professor Namita Singh and organizers for giving me this opportunity to be the part of this wonderful lecture. Really thoroughly enjoyed, though I had some network issue and some of the slides I couldn't see properly, but the narration of the speaker was so good that I thoroughly enjoyed and followed what he was trying to convey to all of us. So coming to Dr. Namit Ranjan, who has given this beautiful talk, wonderful talk on how the translation regulation in eukaryote is affected by the, what is the role of regulatory network and especially the RNA epitranscriptome. So this is a wonderful new idea of understanding epitranscriptome where how the tRNA, RNA, mRNA as well as ribosomes, they are linked. So it's a virtually new concept, and he has shown very beautifully. It took us for about 1 hour in the RNA world. We were virtually in the RNA world, and he has shown how various components they play so meticulously that even one codon can make a lot of sense and can make a lot of differences. And in another part of his presentation, we heard about the role of EF3, which we normally consider so far as a elongation factor, but he showed us that this is actually the translocation factor more over the concept that we usually have in our mind, and he has shown very clearly how using EF2 and EF3, and using latest technologies which people have also questioned how to use them. So using state-of-art new methods available, he has shown. So wonderful lecture, Dr. Namit. I just have a small from my side.
Yes, please. Yes, ma'am, please.
Yeah, Dr. Namit, I have a small query that how the various phenotypes or various diseases, as you mentioned in that they can control various diseases, metabolic disorders, as well as in fact cancer and neuro, how these are regulated by these tRNA modifications. Is it going to be possible just by a change we can convert either to neuro or to cancer. How is that regulated? Is there any information available in the literature of your lab?
So our lab don't have, we don't study this kind of thing because we are mostly biochemists. But the way it is done is that if you take any cancer, for example, let's say diabetes, okay, there when people have seen what are the proteins that are downregulated or upregulated, and there you see that you have tremendous downregulation of tRNA modifying enzymes that results in an unmodified tRNA in the cell, and this slows down translation. And this is the one of the main factors how it can regulate. So it's actually coming from the modifying proteins, which modifies the tRNA, because tRNA is like a very it's a very static molecule, which, let's say, it's a very how do we say, it's a molecule which is but if modifying proteins are not there, the tRNA would not object that I'm unmodified or not. The tRNA would say that, okay, I am there. If you all guys are there to modify me, please modify me, and I'm going to do my job properly. And if there are no guys to modify, which is in the case of cancers, especially many modifying proteins are downregulated, and there are mutations in the genes that is responsible for modifying proteins, and this indeed leads to the cancer. So it's not a direct effect of tRNA which is resulting in disease. Rather, it's actually the upstream players which is responsible for modifying tRNAs. And if you have an unmodified tRNA, you slow down translation, and this would cause the disease. So it's an full circle of feedback mechanism that is going on there. So as a biochemist, we can show that if you have a mutation in certain modifying genes, which would be responsible, or the product from that would be responsible for modifying tRNAs or modifying mRNA, if they are bad, then you result in unmodified mRNA or unmodified tRNA, and that is the cause of disease. Okay.
Yes, yes. Thank you. And thanks again for the wonderful research observations that you shared with us. It's a new concept, and you have wonderfully explained. So
thank you very much.
Thank you. Over to organizers.
Thank you, ma'am. On the behalf of AMI, I thank to Dr. Namit Ranjan for such a wonderful and fascinating presentations and sharing his research with the group and all the participants. On the behalf of AMI, I thanks to today's chairman of the session, Professor Neelam Sangwan ma'am, to facilitate us, to allow us to use this platform for such a wonderful presentation. I'm thankful to all the organizing committee, coordinators, and above all to the technical team from the LUAS for organizing such a wonderful and smooth webinar. And above all, I thanks to all participants who make this webinar possible, and without them, this webinar couldn't be possible. Thanks again, Dr. Namit Ranjan, for such a wonderful presentation. Thank you.
Thank you very much for this opportunity. Thank you.
Okay, thank you very much, Dr. Namit.